Yeah, today class. Now the question is the initiation codon in protein synthesis. What is the initiation codon? The starting codon is called as initiation codon. So in the mRNA, we see the codons. These are also called as triplet codons. Because 3 nitrogen bases form a triplet codon, they are also called as codons. Okay. The first codon in any mRNA, the starting codon, is always AUG. Okay, each codon codes for an amino acid. Okay, methionine is the amino acid coded by AUG. So the starting codon, initiation codon is AUG. But in very few cases, in some of the bacteria, the initiation codon is GUG. So in 99% of the cases, in most of the cases, the starting codon is AUG. In very few cases, it is GUG. So the right answer is AUG or GUG. Okay. So next, when the codon of an amino acid is mutated into a termination codon, it is known as, okay. So the question here is, when the codon of an amino acid is mutated into a termination codon, it is known as nonsense mutation, missense mutation, frame shift mutation, silent mutation. So, there are different types of mutations: point mutations, chromosomal mutation. So, you might have read all this. Here, you see, here there are different mutations. Here, you see, these are the mRNA sequence. This is the mRNA sequence. So, first codon you see here, AUG. Here, in the mRNA, the first codon is AUG. And there are different codons here. In each mRNA, the sequence of the nitrogen bases. These are called as nitrogen. A adenosine. A stands for adenosine. U stands for uracil. Guanosine, cytosine. All these are different kinds of nitrogen bases. And 3 of the in a sequence, that means in a order. How the order of the in which order these nitrogen bases arranged is called as a sequence or genetic sequence. It decides the amino acids which are formed in the protein synthesis, because mRNA acts as a template for the protein synthesis, that is called as translation of mRNA into polypeptide chain or protein. So in the same sequence or same sequence is also called as order. In the same order, the codons arranged in the mRNA, they reflect the order of amino acids which are present in the polypeptide chain of protein. Because see here, the first codon is AUG. So AUG codes for the amino acid methionine. So what is given here? Methionine. And CCG, it codes for the amino acid proline. So this indicates the mRNA chain, and this one below one indicates the polypeptide chain, which, okay. So it is formed by the amino acids in the same order of codons in the mRNA. So here you see, this is the unmutated mRNA sequence, and from this, in the protein synthesis, here polypeptide chain is produced with the sequence of methionine, proline, tyrosine, valine, proline, like that. And there is one codon that is called as stop. Here it is indicated by stop, that is called as UAA. UAA is one of the termination codons. You might have known there are 3 termination codons. These are also called as nonsense codons. That means, see, in the mRNA, there are 64 types of the codons are present. That is called as genetic code. In the genetic code, there are 64 types of the codons. Out of the 64 types of the codons, only 61 codons code for the amino acids. The remaining 3 codons do not code for any amino acid. Here you see CCU, it codes for proline. But next there is UAA. This UAA is a nonsense codon or termination codon. It does not code for any amino acid. So it is called as nonsense because this codon doesn't make any sense. It doesn't code for any amino acid. So protein translation ends here. It is stopped. So this codon is called as termination codon because whenever this codon is present, that protein synthesis is terminated. So that you should understand what are the termination initiation. Since we already discussed about the initiation codon or starting codon, we should also know about the termination codons or nonsense codons. There are 3 types of the nonsense codons: UAA, UAG, UGA. Okay, so that is the general information here I discussed. And now we come to the mutations part. There are different types of the mutations. Now we are discussing about the point mutations. What is point mutation? One of the nitrogen bases of the codon is replaced by the another nitrogen base, that is called as point mutation. It is changed. It is replaced by other. So here you see what is silent mutation? See AUG, CCG, UAU, EAC, GUU. Here this is the original mRNA, and now we are seeing in the silent mutation, instead of the A, that is adenine in the GUA codon, here A is replaced by U here, uracil. Adenine is replaced by uracil here. But the thing is, GUA codes for the valine, and even though GUA is mutated to GUU, but it also codes for the same amino acid. That means it doesn't make any difference with respect to the polypeptide chain sequence. Same order of amino acids is produced even though there is a mutation. There is a replacement of A by U here. It codes for the same amino acid because there are each amino acid codes by, it is coded by more than one codon. Many times it occurs because here you see valine is coded by, valine is the amino acid, it is coded by GUA and also by GUU also. So though there is a replacement of adenine by the uracil in the mRNA, there is no change with respect to the amino acid sequence here. Same amino acid, it codes for the same. So this mutation is called as silent mutation. It is called as silent mutation because there is no change with respect to the sequence of the amino acids in the polypeptide chain. And next, there is one more type of the mutation. Here you see where there is a change here, yeah, AUG, and here instead of CCG, here what we see, cytosine is replaced by uracil here, and it codes for different amino acid. Unlike in the case of the silent mutation here it is different kind of the mutation. The C is replaced by U here, and it codes for different types of the amino acid, that is serine. So instead of the proline in the second position, serine is present. That means the amino acid sequence is changed in the polypeptide chain. So this mutation is called as missense mutation. Okay, so there is a change in the silent mutation. There is no change of amino acid, but in missense mutation there is a change in amino acid. And one more thing is the question we are discussing now, the nonsense mutation. In the nonsense mutation, what happens here? AUG, CCG, UAU, UAA. Here what happens? UAC codon here changed into UAA because C is replaced by A. Cytosine is replaced by adenine. But the thing is, UAC codes for tyrosine, but UAA, as we discussed earlier, it is a stop codon or nonsense codon, termination codon. It doesn't code for any amino acid. Okay, so it is a UAA is a nonsense codon. UAC after mutation changed into UAA, that is a termination codon or nonsense codon. So this mutation is called as nonsense mutation because the protein synthesis is stopped. It is terminated here by the formation of the nonsense codon here due to the replacement of the C by A in the mutation, the point mutation. That polypeptide chain is terminated abruptly. So this mutation is called as nonsense mutation. So you should remember this. And there is one more kind of the mutation that is called as frame shift mutation. What is frame shift mutation here? ATG in the DNA. ATG. You should you can easily find whether it is mRNA or DNA. If U is present in the sequence, you can recognize it as an mRNA. But here, instead of U, here in the example, T is given. That means in the DNA, the mutation may occur at the DNA level or at the mRNA level also. So here what I have shown, ATG, CAT, TGC. So these are three codons here. ATG is one, next codon is CAT, next codon is TGC. But here, due to the frame shift mutation, what is frame shift mutation? Because ATG, between the T and G, another nitrogen base, it is incorporated in between the T and G. So it changed the codon. It shifted the codon from ATG to ATT. So because one additional nitrogen base is incorporated in between T and G, so now instead of ATG, the codon is ATT, and the entire codon sequence is shifted from now onwards. Instead of CAT, it became GCA. Next, instead of TGC, it became TTG. So there is a shift in the codon. Entirely, all the codons are changed. So it forms a drastic change just by the incorporation of the one nitrogen base in between. The total codon, all the codons are shifted, changed. So this is called as frame shift mutation. This is called as frame shift mutation. So you have to remember all these types of the point mutation: silent mutation, missense mutation, nonsense mutation, and frame shift mutation. Now we see the question is, when the codon of an amino acid is mutated into a termination codon, it is known as, what is this? Is nonsense mutation, because it formed a nonsense codon. So I already told you, UAA, UGA, or UAG. These three are called as nonsense codons. When these codons are formed, that mutation is called as nonsense mutation. Okay, next. The rearrangement of some genes in reverse order in a chromosome. Okay. So in the chromosome, the rearrangement of some genes in reverse order in a chromosome is. Okay. So there are different kinds of the mutations, I told you. Inversions, deletions, duplications, and translocation. So these are different kinds of the mutations here. I think, Rajia, you have to remove these lines by yourself. You have to erase, use the eraser to remove this. It is not here. Okay. So Rajia, remove these lines, okay, by erasure. So now we see different kinds of the chromosomal mutations. Earlier we discussed about the point mutations at the nitrogen base level, at a small level. But here we see the mutations at the chromosomal level. These are also called as structural mutations at the chromosome level. So here what you see, inversion. What is the inversion? Here you see there are genes A, B, C, D, E, F, G, H, I. These are different genes present. Here the chromosome, there is a break here at the two places. Chromosome is broken at two places. So there is a small, there is a part of the chromosome. It is deleted here from the chromosome. And what happens here? The same fragment of the chromosome which is deleted is again, it is inverted, and again it binds to the same chromosome but in a reverse order. Okay, the same fragment of the same chromosome, it binds in the same place but in a reverse order. Instead of DEF, here what we see after inversion, FED. This is called as inversion. Okay, and next one more thing is deletion. Here, just like earlier case, here there that chromosome is broken at two places, 2 points, and a part of a fragment of the DNA is deleted, but it is deleted permanently. It doesn't binds again. It is permanently deleted. So now you see, instead of A, B, C, D, E, F, G, H, I genes in the order, what we see here, A, B, C, next G, H, I. That means DEF fragment is deleted permanently. So that is called as deletion. In inversion, the deleted part again binds back in the same position, same place, but in a inverse order, reverse order. But here in a deletion, it is totally deleted. So that is one type of the chromosomal mutation. And another one is duplication. Here you see in the chromosome, a part of the fragment is duplicated. GHI, this part is duplicated. So here, additional to the normal chromosome, here the extra part of the same, this FGH, is duplicated. This is called duplication. And one more thing is translocation. Here in the translocation, what happens in the homologous chromosomes? A part of the chromosome is deleted here from this chromosome, but it binds to the another chromosome, homologous chromosome. You see, A, B, C, D, E, F, it becomes just A, B, C. A part of the fragment is deleted here. It is broken. D, E, F part is broken. And it binds to another chromosome. This is called as translocation. So you have to remember what is inversion, deletion, duplication, translocation. Now the question is about the inversion. So what is the question? It is called as vilomam antaru Telugulo. The rearrangement of some genes in reverse order in a chromosome is called as inversion. If that part is totally deleted, that is called deletion. Duplication means extra part. Some part of the chromosome is duplicated and it binds to the same chromosome. But the deleted part binds to the another chromosome, that is called as translocation. So all these you have to remember. Next one is the hereditary disease restricted to males and characterized by excessive bleeding is. Actually, it comes under zoology usually, but they asked this question in botany. So anyway, see, the hereditary disease restricted to males. Okay, that means there are males means it is sex linked disease. In the males we find XY chromosome. In the females we find XX chromosome. Okay, if there is any mutation in the X chromosome, what happens? If that is a recessive mutation, it is not reflected in the females. Mostly it is reflected in the males only, because in the males there is only single X chromosome. If there is any mutation, that male is affected immediately. Most of the times, if there is any mutation in the X chromosome, usually females act as the carriers. That means the disease is not expressed in the females, but it is expressed in the males. In most of the times, it is expressed in the males only. So it is called as sex linked. So here, what is the excessive bleeding means? There is no, if there is any problem, there is no clotting. Okay, there is no clotting of the due to this disease. So this disease is called as hemophilia. So there are different kinds of the diseases. These are sex linked. That is hemophilia is the answer for this. And one more disease, sickle cell anemia. That is not the right answer, but you should remember that also. What is sickle cell anemia? That means that RBC, it becomes sickle shape. And albinism means that person, they turn to white color. Their skin, okay, they become whitish, and hair lip. So these are the different kinds of the diseases. See, we see now different diseases just for the purpose of the competitive exams. So there are different types of disease, like syndromes. These are also called as syndromes. Down syndrome. You know what is Down syndrome? That is the extra chromosome, 21 chromosomes. Usually there are 2 chromosomes, a pair of chromosomes. There are 23 pairs of the chromosomes in the human beings. Each one have pair, okay? First pair, second pair, third pair. So this is the 21st chromosome. Usually there are 2 chromosomes in the form of the pair, but there is one more additional chromosome, that 21, that chromosome. Here we see instead of 2 chromosomes, we see 3 chromosomes. That means there is one extra chromosome. That is called as trisomic condition. That is also, we can call it as 2n plus 1 condition. 2n plus 1 condition. 2n means 46 chromosomes. If it is 2n plus 1, they have 47 chromosomes, one extra chromosome. So 21st chromosome, it is extra. That is 2n plus 1 condition. Here what we see, that is called as Down syndrome. Okay, it is not sex linked disease. Okay, since these are called as autosomes or allosomes. Autosomes means they are not sex linked. X and Y chromosomes, these are called as allosomes. They are sex chromosomes. Remaining are called as autosomes. So it is not sex linked. So Down syndrome can be seen in both the males or females. And the next one is Klinefelter syndrome. Here it is again sex linked. Male with extra X chromosome, only males. In the Klinefelter syndrome, if the male has one extra chromosome, instead of XY, they have XXY. That is called as Klinefelter syndrome. It is seen only in the males. It is sex-linked syndrome. Tall stature, low testosterone, infertility, gynecomastia. What is gynecomastia? The formation, development of breast in the males. So this syndrome is called as Klinefelter syndrome. There is an extra X chromosome. And Turner syndrome. Here it is restricted to females only. Turner syndrome. It is also X-linked. Here there is only one X chromosome. Instead of two X chromosomes in the female, if there is only one X chromosome, that is shown as XO. So that syndrome is called as Turner syndrome. And sickle cell anemia, it is not sex linked here. But what happened here? This is an autosomal recessive problem. The normal RBC, okay, the morphology, the shape of the red blood cell is shifted to sickle shape. That is called as sickle cell anemia. Okay, because RBC, since it is changed its shape, there is a problem of carrying oxygen. Okay, so this is called as sickle cell anemia. Anemia means deficiency of RBC, blood cells. And hemophilia, what is hemophilia? Just the question now we are discussing is about hemophilia. What is the hemophilia? Blood does not clot. Okay, if there is an injury to a person, there is a continuous bleeding because blood cannot clot properly due to this disease. It is X-linked disease and it is mostly seen in the males only because that mutation happens in the X-linked X chromosome only. It is a recessive. That means if both the X chromosomes have this mutation only, that disease is expressed in the females. But in case of the males, even in one X chromosome has the mutation, that hemophilia is shown. It is expressed in the males because males have a single X chromosome. So mostly this disease is seen in the males only. Females act as the carriers to the next generation of the males. So hemophilia. Okay, next, that is the right answer to the question we are discussing. Next, one more disease, color blindness. That is, cannot distinguish certain colors like red color, yellow color, green color. These colors cannot be distinguished. They also don't know that unless there is one test. Usually in army, they test this one by one chart here. So it is X-linked recessive. Again, this color blindness also, it is mostly seen in case of the males only. Phenylketonuria, this is one more disease related to chromosomes. Body cannot digest phenylalanine. Phenylalanine is the amino acid. Okay, so if they have phenylalanine, they have problem because that deficiency of enzyme phenylalanine hydroxylase. This phenylalanine amino acid is not metabolized due to the deficiency of this enzyme due to this mutation. Okay, so this phenylketonuria. So all these are different kinds of the syndromes and disease which are mostly related to X chromosome and sometimes autosome. Okay, you remember this. Next. When so the right answer to the question is hemophilia, the hereditary disease restricted to males and characterized by excessive bleeding. That is hemophilia. Okay. Next, when organisms lack one individual chromosome, it is known as. Already we discussed. In case we have normal chromosomes is shown as 2n. If there is one chromosome is less, it is called as 2n minus 1. If there is one chromosome excess, it is called as 2n plus 1. Okay. So here, what we see here, numerical changes in the chromosome. These are all mutations only. Chromosome mutations at the chromosomal level. Chromosome euploidy, aneuploidy. Euploidy means, see, monoploidy. If there is a single set of chromosomes, usually in the gametes, we see a single set of chromosomes that is shown as X or N. Monoploidy, a single set of chromosomes. Diploidy, after the fertilization in the zygote, we see diploid condition. That means two sets of the chromosomes, one from the male parent, another is from the female parent, another set. So there are two sets. A polyploidy, what is polyploidy? If there are three sets of the chromosomes, that is called as triploid. If there are four sets of the chromosomes, that is called a tetraploid. If there are six sets of the chromosomes, which is called as hexaploid. So this is all euploidy. Okay. Next, aneuploidy. That means aneuploidy means the change occurs at one or two number of chromosomes. If one chromosome is less, you see, for example, human beings have 46 chromosomes. 2n is equal to 46. If there is one chromosome less, that is called as 2n minus 1. Instead of 46, if they have 45 chromosomes only, that is called as monosomy. 2n minus 1. Nullisomy. If they don't have two chromosomes, that means instead of 46 chromosomes, if they have 44 chromosomes only, that is shown as 2n minus 2. That means 40 minus 46 minus 2, that is 44. Nullisomy. And trisomy, here it is called a hyperploid. That means chromosomes are excess here. Hypoploid means chromosomes are less than the normal. Hyperploid means chromosomes are more than the normal. Trisomy, that means 2n minus plus 1. If it is minus 1, 45 chromosomes. If it is plus 1, 47 chromosomes. Tetrasomy, two chromosomes are extra here, opposite to nullisomy. Okay, tetrasomy means 2n plus 2, 48 chromosomes. So you have to remember all this. So the right answer to the question is monosomy. If the one chromosome is less, when organisms lack one individual chromosome, it is known as monosomy. The right answer is monosomy. Next question is, spindle fibers are formed by. Where we find the spindle fibers? During the cell division. During the cell division, we find the spindle fibers. But in the normal case, we don't find spindle fibers. So in normal condition, when there is no cell division, they are in the form of the microtubules. And during the cell division, all these microtubules are polymerized. One after one, all these microtubules are joined to form a thread-like structure that is called as spindle fiber. Okay, so spindle fibers are formed by microtubules. Okay, in the resting period, when the cell is in the resting period without cell division, spindle fibers are changed into microtubules. Okay, so next question is, kappa particles in Paramecium aurelia was reported by. Usually Paramecium, they don't give the questions from the Paramecium. Usually it is treated as an animal. So anyway, since that question is given, you see here, this is the Paramecium here and slipper shape. And here, this Paramecium has some dot-like structures which are called as kappa particles. These kappa particles were discovered by Sonneborn, okay, Sonneborn in Paramecium aurelia, and later they were they found as the endosymbiotic bacteria, those particles, and they release a toxic material, toxin, that is called as paramecin. The toxin produced from the paramecium is called as paramecin. This paramecin, how it is produced? From the kappa particles. Okay. So those paramecia with the kappa particles have an advantage because those toxins kill the paramecium without kappa particles. Okay, so kappa particles give an advantage to the paramecia to act against the opponents. Okay, so the right answer is Soneba. Kappa particles in Paramecium aurelia was reported by T.M. Sonneborn. That is the answer. Next, you see there are different. You see here, Daves and Danieli. Yesterday we discussed trilamellar or sandwich model of the plasma membrane as discovered by, explained by Daves and Danieli. That also you have to remember. Next, another question. Genes which result in the death of the individual carrying them are called. These are called as lethal genes because the genes are responsible for the death of that organism. These are called as lethal genes. That is the right question. Right answer. Next, photolysis of water results in the evolution of. See where this photolysis takes place. That you see in the photosynthesis in the light phase, we see in the non-cyclic photophosphorylation there are two pigment systems, PS1 and PS2. So here at the PS2 there is one oxygen evolving complex. It is also called as OEC. Okay, in the non-cyclic photophosphorylation, you see there are two photosystems, photosystem 2 and photosystem 1, and the reaction center is called as P680 in the PS2 and P700 in case of the PS1. Here oxygen is released from the H2O. H2O is split in the presence of the light only, and this oxygen which is released is the one is responsible for us. Okay, we are able to live only because of this oxygen released in the photolysis of the water. Okay, the oxygen which is released from the plants in the photosynthesis, from where it is released in the light phase at the PS2. Okay, that you have to remember. And photolysis of water results in the evolution of O2, oxygen. The number of chromosomes in Arabidopsis thaliana. How many chromosomes are present? So there are 5 here it is given as 5 chromosomes, but actually 5 pairs of chromosomes or 10 chromosomes is the right answer. But since it is not given as 10 chromosomes, we can give the answer as 5 chromosomes. Actually, the number of chromosomes in Arabidopsis thaliana are 10. Okay. And microinjection of DNA is transferred by, what is that method? Direct method of transfer. And uracilin, I think we are running short of the time. You see here direct methods, gene gun method, microinjection, electroporation, electric pulses, all these are called as direct methods. And agrobacterium mediated gene transfer is called as indirect method. We can use the vectors like TA plasmids. This is the indirect method of gene transfer, and direct methods of gene transfer are